Hello, my name is Rama Ranganathan, and I'm a professor in the Green Center for Systems Biology and also with the Departments of Biophysics and Pharmacology at UT Southwestern Medical Center. In the next 3 lectures, I'm going to tell you about a problem that I've been very interested in for many years now, and that is the problem of so-called evolutionary design of proteins. So what we're going to do in the next 3 lectures is to begin with a general overview and a statement of the problem. What do we mean by this problem of the design of proteins? And what's the nature of the problem? Why is it a difficult and interesting problem? In the second lecture, we will move on to present or talk about an approach to this problem that we have started in our laboratory and discuss a general model for proteins that emerges from these methods. And in lecture 3, we'll talk about experimental approaches for testing the model and ultimately producing a general hypothesis that will drive future research. So let's begin with a statement of the problem. What do we mean by evolutionary design of proteins? So the problem of proteins, in some sense, is very easy to state, though difficult perhaps to solve. And that is that we know that proteins are synthesized as these long polymers of basic building blocks that we call the amino acids. And classical studies teach us that information in the sequence is such that if you put this polypeptide into a physiological buffer, it is able to spontaneously fold up into this precise and well-ordered 3-dimensional structure. That problem we call the folding problem of proteins. But then since evolution is designing these sequences not for just the property of folding, but for biochemical functions, there's information written in the sequence not just to specify the folding problem, but the capacity of the folded state to carry out various biochemical activities. So for example, in the case of this little protein, the PDZ domain, the ability to recognize target ligands, small peptides, at a groove on the surface that we call the peptide binding pocket. So fundamentally, the question is the nature of this information encoding problem. What information in the sequence, what constraints on amino acids and in principle interactions between amino acids is necessary and sufficient to produce these properties of proteins, the capacity to fold, the capacity to carry out biochemical activities. And I want to point out that natural proteins have to do in principle more than just fold and carry out biochemical functions. They have to be possible through the process of evolution and able to be to have the capacity for adaptation, to evolve to new functions when the nature of the fitness function demands it. So we're interested in understanding what design of proteins accounts for all of these properties. Now, what do we actually mean by this word design? What exactly is meant by design? Well, first thing to realize is all the properties of proteins that we're interested in, folding, biochemical activities, and even this property of adaptation, arise somehow from the pattern of physical interactions between all the atoms that make up the structure. So by design, we specifically mean this pattern, the architecture of physical interactions between atoms, and in principle, the generative process, as say evolution, that shapes this pattern. And we can gain some intuition on this problem of the pattern of these physical forces by knowledge of physical chemistry. For example, we know that all the fundamental forces that bind atoms act at very short range, and they act with great distance and geometry dependence. As a consequence, when we look at atomic resolution structures of proteins, we observe this property of good packing. And let me explain what this means. So what I'm showing in this images is a slice through the core of this protein. So I've chopped this protein down the middle and opened it up like a book. And you can look at the way that the atoms are arranged in the core of this protein. And what we observe is that the density of atoms in the core is so high that it approaches that which we observe in crystals of free amino acids. And as a consequence, we find that proteins look in some sense like 3-dimensional jigsaw puzzles. That is to say, they look precise and locally exact. As a consequence, when we ask questions about proteins, questions about protein function, such as which amino acids in this protein are responsible for recognizing this substrate ligand in yellow, we all use a principle, I would call it the principle of spatial proximity. What that means is that, well, if you want to know which amino acids are recognizing this peptide, the idea is to look in the immediate environment of that peptide in the structure. So the red amino acids are the ones that are physically contacting the substrate ligand, and a first-order hypothesis would be that those amino acids are the ones that are responsible for the specificity and affinity of target ligands for this PDZ domain. And in fact, many experimental and computational studies are done to interrogate this local environment for the origin of binding and specificity. But the trouble is that even in this little protein, there are backside surfaces. So for example, this patch of red that's in the back of this protein, quite far away from where its substrate ligands are binding at the active site, but these surface sites control the affinity and specificity of target ligands for the active site. And what's interesting about those kind of surface sites that have this capacity is that they're quite special. There are many other surface sites that you can see in blue that are equidistant and just as well connected to this pattern of interactions to the active site, but they're inert. They don't have this property. Interactions there do not affect the affinity and specificity of ligands at the active site. This already tells us then that there must be some heterogeneity, some pattern of interactions which we don't observe in the structure of the protein that makes it so certain surface sites are functionalized and can carry out these properties of long-range communication, whereas many other surfaces do not have that property. And that's not obvious when we look at the protein structure. Now, this property of long-range interactions is not just an arcane property of this one protein. In fact, one can argue that biology in general is driven by such long-range interactions. For example, in membrane receptors such as G protein-coupled receptors, signaling proteins like G proteins, and other receptor molecules such as the TUNB-dependent receptors that are in the outer membrane of bacteria, there is a critical need for long-range interactions. So for example, in G protein-coupled receptors, when ligands bind at an extracellularly accessible site over there, the effect of that binding is felt at quite a long range in cytoplasmic regions that mediate downstream signaling. And the fundamental purpose of these proteins is to convey this information from one site to a site that's quite at a long distance. It's not just signaling proteins. Even it's now clear that in enzymes, active site function can depend upon the dynamics of surface loops that are quite far away. So we would like to say generally that long-range interactions are important in biology. They mediate signal transmission, catalytic specificity, and efficiency. And these are often defining features of protein families. So these long-range interactions tend to be quite important. This problem of trying to understand long-range interactions is also not anything new. In fact, it dates back to the origin of structural biology and our understanding of proteins. So for example, this is the classic problem of understanding function in hemoglobin. Hemoglobin, of course, was a structure was solved by Max Perutz, shown here, in the late 50s. And the problem of hemoglobin was well known even at that time. Hemoglobin comes as a heterotetramer of alpha and beta subunits, and the structure of the molecule is shown here. Each of these subunits contains a heme moiety that ligates an oxygen atom. And the interesting property of hemoglobin that was known since the beginning of the last century is cooperativity in oxygen binding, such that an oxygen ligating to one of the heme molecules influences the affinity of oxygen for a distantly positioned heme on a different subunit. And of course, when the structure of hemoglobin was done, the first question was, how does this occur? How does cooperativity in oxygen binding emerge from properties of the three-dimensional structure? Now, to illustrate the nature of this problem, I'm actually going to let someone else tell you the nature of the story. It turns out that the great physicist Richard Feynman in the early 60s gave a lecture called The Relation of Physics to the Other Sciences, which actually was recorded. And in this lecture, he addresses this problem of hemoglobin. And I'm going to play a short audio clip that, according to Feynman, illustrates sort of the nature of this problem. So let's listen to Richard Feynman, and we'll come back in just a minute.
One of the great triumphs of recent times within the last year, in fact, was at last to discover the exact arrangement of space and the order of all the amines, some 56 or 60 amino acids in a row, over 1,000 atoms, well, nearly 2,000 if you count the hydrogen atoms, have been all located in a complex pattern for one, well, now two proteins. The first was hemoglobin. One of the sad parts of this is that as far as we can tell yet, we can't see anything from the pattern. We don't understand why it works the way it does because of that pattern, but of course that's the next problem.
Okay, so this is 1961, and Feynman claims that this is the next problem to solve. The understanding how this pattern, the idea was we finally have the structure, the precise position of all the atoms that make up hemoglobin, but we can't see in this pattern how important functional properties such as the cooperativity in oxygen binding emerges from the structure. I'll point out that Feynman gets many things wrong here. I mean, hemoglobin doesn't have 55 or 60 amino acids, but like every good scientist, he really gets the essence of the problem. We don't understand the function of the protein given the structure. And the question really is why? What's the fundamental problem that limits our ability to understand protein function given the structure of proteins? And I'll sort of summarize the whole thing by saying that the essence of it is that we don't see energy in structures. And what I mean by this is that we may observe in this molecule say a hydrogen bond that relates two of the atoms, but what is the net value of this hydrogen bond in this complex transaction of free energies that happen between the unfolded state of the protein and the folded state? In fact, everything we know about that problem teaches us why it's such a difficult thing to know the net value of these interactions in protein structures. First of all, natural proteins tend to fold to very marginally stable, to very marginal stability. So for example, a typical molecule such as this, the PDZ domain, might be stable by just 8 or 10 kilocalories per mole. That separates the unfolded state and the folded state. And what does 8 or 10 kilocalories per mole mean to you and me? Well, that's on the order of 3 or 4 good hydrogen bonds worth of free energy distributed through all of the intraatomic interactions in this protein structure. And also coupled with what I told you earlier, which is that the forces that bind atoms act with such distance and geometry dependence, it's no wonder that by looking at protein structures, we don't know the net value of all of these interactions. And that is really the essence of the problem. Which of these interactions between atoms are important and which are not so important for specifying properties of the protein, folding, function, and in principle, properties such as adaptability. Well, how do we solve this problem then? We know some things about the nature of the solution, though, that can help us, that can guide us in looking for an approach. And I'll tell you about 3 of these sort of nature of the solution that I think that the empirical data collected over many, many years tells us. We're looking for a solution that actually has 3 characteristics. The first characteristic, which is interesting, is that it's sparse. And what I mean by that is that most of the important contributions to folding and function in many proteins are contained within just a small fraction or a subset of the amino acids that make up the protein. One way to show this result is to use mutagenesis methods to probe the importance of amino acids for, let's say, protein function. So for example, what I'm showing here is the thing I showed you earlier, the structure of the PDZ domain sliced into the core, showing that it looks like a homogeneous and dense network of amino acid interactions. But if you carry out a comprehensive mutagenesis study, so in this matrix, what's shown is the effect of mutating every position in this protein, going from N-terminus to C-terminus on the rows of the matrix, to every other amino acid on the columns of this matrix. And every pixel of this matrix reports the functional effect of that mutation on the ability of the PDZ domain to bind substrate ligand. And the result is quite striking. You see that there are some positions, such as this one, that wants to be the native amino acid, which is histidine, and essentially can't tolerate any mutation. Any mutation at that position is deleterious for the binding of the substrate ligand. Yet there are other places, like the amino acid just next door, which could care less what you put at that position. Every amino acid suffices to give you native-like function. If you examine this pattern of amino acid importance over the whole sequence of the protein, what you conclude is that there's just about 20% of the amino acids that are critical for the property of protein binding. And actually, I'm not showing it here, but if you look on the protein structure, it includes amino acids that are both near and far from the site at which peptides bind the PDZ domain. So this is really a data set that illustrates the sparsity and heterogeneity of proteins in their function, which is not obvious when we look at the protein structure. The second property is more complex to explain, and that is that the solution is non-additive. That is that amino acids that are involved in function are often not acting independently, but they're engaged in pairwise or even higher order cooperative interactions, such that the effect of the amino acids taken together is not just the additive or the independent actions of the amino acids taken independently. That requires a little bit of explanation because it's not a simple thing to understand. So I'm going to review very quickly the concept of thermodynamic cooperativity and what it means to say that amino acids are acting non-independently, because this is quite a critical problem in our understanding of proteins. So what I'm showing you here is just a model protein, a schematic of a protein. I've labeled 3 of the amino acids. So consider this protein with these 3 amino acids. How can we sort of generally describe the effects of mutations at these positions with regard to some equilibrium free energy measurement that we make on the protein? And what I mean by that is we might observe or measure the effect of these mutations on, say, the stability of the protein, or perhaps on the ability for it to bind substrate ligands or any other equilibrium free energy measurement. So how can we generally describe the effect of these mutations on such a measurement? The single mutation effect is simple to describe. You measure the free energy of the wild type state, you measure the free energy of the mutant state, and take a difference between the two, and that is the energetic effect of a single mutation on whatever property is you're measuring. But the effect of a double mutation is more complicated. In this representation, the formal description of how to think about a double mutant experiment, what we do is we make what's called a thermodynamic box, or a Carnot cycle. And the idea is that each corner of this box represents one state of the protein. So this is the wild type protein. This is the effect of mutating position I. On this dimension, we put the mutation of position J, and this corner now has the double mutation state, where both positions are mutated jointly. And the idea is that we're trying to write an equation that describes most generally the energetic effect of the double mutation. That's the difference between this state and the diagonal state, which is the wild type. That's the effect of the double mutation. So it turns out that in general, the right way to think about it is that the effect of the double mutation is the summed effect of the 2 single mutations, but corrected by a factor called the coupling free energy between the 2 mutations, right? And the idea is that if the 2 mutations are totally independent of each other, they are thermodynamically independent, then the coupling free energy is 0. Then it's simple. The effect of the double mutation is just the additive effect of the 2 single mutations. But if the 2 mutations see each other, if you will, or are interacting with each other, then there's a coupling free energy that must be taken into account in describing the energetic effect of a double mutation. Now, what is this coupling free energy? Well, it's interesting when you look at how it's defined. The effect of the coupling free energy, or the delta G between mutations I and J, is the energetic effect of making a single mutation in the wild type background minus the energetic effect of that same mutation I, made in the background of another mutation J. Okay? So to be clear, this quantity, the coupling free energy, can be thought of as sort of 2 things. They're equivalent. The first is the degree to which the effect of one mutation depends on the background of another mutation. And of course, if the effect of the first mutation is entirely independent of the other mutation, these 2 quantities on the right side of this equation are the same, and the coupling free energy comes out to be 0. That is, if 2 mutations are thermodynamically independent, the coupling free energy is 0. But if it's non-zero, then that means those 2 mutations are interacting with each other, and the magnitude of this coupling energy measures the degree of coupling between the 2 mutations. Okay, that's the formal description of a double mutation, but we're not done yet. Why stop with 2 mutations? What if we may introduce now a 3rd mutation? How should we think about that? Well, now the problem expands geometrically. So now we have to describe a triple mutation experiment formally in thermodynamic terms as a cube, right? Where the front face of this cube is precisely the same thing that we have here. It's the cycle that describes the energetic effect of a double mutation. And now the 3rd dimension going into the board is the effect of the 3rd mutation, okay? And I won't bother describing the details of the calculation of how to write down the energetic effect of the triple mutation. See, the triple mutation effect is the cube diagonal, the effect of this state minus the effect of the wild type state here. And without derivation, I'll just make a simple point, which is that formally, to describe the energetic effect of a triple mutation, one has to consider the additive effect of the 3 single mutations corrected by all the underlying pairwise couplings, that is to say, the coupling between mutations I and J, the coupling between mutation I and K, and the coupling between mutations J and K. And then there's a new quantity that emerges, which is a three-way coupling, a delta, delta, delta G, which is the degree to which all three mutations interact, which is not simply the additive effect of their underlying pairwise interactions. So you can see where this is going. If you consider 4 mutations, you can imagine what's going to happen. We're going to have to introduce, at least formally, a fourth order term that describes the coupling between all 4 mutations and so on and so forth. So this becomes an extremely complex problem when one considers the idea that a typical, even small protein is made up of maybe 100 amino acids. So the question we can ask is, in practice, in real proteins, do we observe such high-order thermodynamic couplings? And really, what is the extent of the problem of cooperativity between amino acids in practical cases? I mean, we don't know much about this, actually, because it turns out to be very difficult to measure these high-order energies, but it has been done beautifully in a study in the potassium ion channel. So let me just describe to you what these investigators saw. And here I'm talking about specifically the work of Sadovsky and Yifrak that was reported a few years ago. So just to introduce the model system they were working on, this is the potassium ion channel. This is a membrane protein that's found in the plasma membrane of many cells and is evolved in selective permeation of potassium ions across the plasma membrane. The structure of the potassium channel is shown here. It's actually a tetrameric protein, but only 2 of the subunits are shown here for clarity. This is the outside of the cell and the inside of the cell. So the channel adopts a roughly inverted TP-like configuration, where on the outside face of the channel, there's a narrow constriction right at the top that's called the selectivity filter. That's a narrow region where potassium ions go through the pore in a single file. And then on the internal aspect, near the cytoplasmic face, there's a narrowing of these helices in a region that's called the activation gate, and that region has to undergo a conformational change to allow the channel to open to conduct ions. Now, what was really interesting about the work from Sadovsky and Yifrak is that they showed that amino acids in the channel that are important for gating, that is, the ones that influence the ability of the protein to open and close, actually make up a network of amino acids that connects the activation gate down at the cytoplasmic region with the selectivity filter that's located more near the extracellular region of the channel. And experiments these investigators did in actually measuring second order, third order, and even fourth order coupling free energies between these mutations showed that along the pathway, and the pathway, by the way, is indicated here in these red amino acids, along the pathway, amino acids tend to show strong higher order terms. That is to say that there are fourth order coupling energies, and they didn't go any higher than that, but maybe there are even higher order terms there. But what's interesting is that though along the pathway you see these higher order terms accumulate, if you go orthogonal to the pathway, that is to mutate residues that are leading away from the pathway, this cooperativity degrades rapidly, and now amino acids are only engaged in low order cooperative terms, or not even cooperative at all. So what this really teaches us, or suggests to us, is that inside of this protein is embedded some cooperative subsystem of amino acids that makes it possible for information to flow from 2 distinct points of the protein. And outside of this system, amino acids are engaged in much more low-order, simple energetic interactions. This is a very illustrative example of the idea that proteins do contain non-trivial non-additivities, that is, say, cooperative interactions of amino acids, although that study also suggested that those cooperativities were loaded perhaps in only a small networks of amino acids and not so involved in the majority of the protein. But that's a statement that requires much more study. To review what we said so far, again, we're looking for something about characteristics of the nature of the solution to the pattern of physical interactions that are in the protein structure. And we find that there are these 3 characteristics. The first is sparsity. That is to say, most of the contributions of amino acids is contained in a small fraction of the residues. The second is this property of non-additivity. What I've shown you just now is that at least the functional residues in the protein are often engaged in pairwise or even higher order cooperative interactions, although the work that I showed you just now suggests that these non-additivities may be localized in just a subset of amino acids that are functionally important, with less importance of this cooperativity in surrounding amino acids. But those assertions will need to be tested further. In addition to these two properties, the third property that we think about is the property of modularity. And what modularity means is that different groups of amino acids can encode for different and independent properties of the protein. And an example of this was illustrated in the study of these proteins known as the S1A serine proteases. These are the enzymes that catalyze the hydrolysis of peptide bonds and involved in many things in biology. But the analysis of this protein family showed that this single protein contains three distinct functional units, the red cluster, the green cluster, and the blue cluster. And experimental studies showed that these clusters mediate distinct and orthogonal biochemical properties of the enzyme. So the important point is that, that these properties of proteins, sparsity, non-additivity, and modularity, in general, are not evident in our examination of even high-resolution atomic structures for all the reasons that we've said so far. And so what we need to ask is, what approach can provide insight into this pattern of constraints? What's an approach that can lead us to the analysis of the so-called design of natural proteins? In the next lecture, I would like to propose an approach to this problem that we've taken in my group, and then discuss a general model that emerges from these studies. Thank you very much.